So hi, today in Biochemistry Bits, we are going to discuss a very short topic, glycogen metabolism. So what is glycogen? Glycogen is the storage form of glucose in case of animals. So it is a homopolysaccharide. So regarding its metabolism, we are going to see in two different pathways. One is glycogenesis, which is the synthesis of glycogen, or this is the anabolic pathway, how we synthesize glycogen, glycogenesis. The other pathway is glycogenolysis, which is the breakdown of glycogen, how we break down glycogen to form glucose. So these two constitute the glycogen metabolism. So now we have an overview of the glycogen metabolism. The most common organs where it occurs is liver and muscle. The need for this particular pathway is synthesis of glycogen to store glucose in case of animals. Now the left side of this picture is the glycogen synthesis pathway or the glycogenesis. The right side is the glycogenolysis or the glycogen breakdown. So let us start with glycogen synthesis or glycogenesis. Glucose is converted to glucose 6 phosphate. We need to trap glucose in the form of phosphate. So glucose will be converted to glucose 6 phosphate. Enzyme is hexokinase in muscle and glucokinase in liver. Now the glucose 6 phosphate will be converted to phosphoglucomutase to glucose 1 phosphate. Now we need to activate the glucose. To activated form of glucose is UDP glucose. It has to combine with UDP. So here the enzyme is UDP glucose pyrophosphorylase. Now a lot of UDP glucose molecules are there. We need to store it in the form of glycogen. So for that you need a core structure which is known as glycogenin. Glycogenin is a protein. This protein is glucosylated on the tyrosine residue. Wherever there are tyrosine residues, you have a glucose residue attached to it. Okay. Now, a lot of glucose residues are there. Glycogenin itself acts as an enzyme and takes up 7 UDP glucose and adds the 7 glucose residues to the glycogenin protein. So, glycogenin acts as an enzyme and catalyzes the transfer of 7 glucose residues from UDP glucose. Now, the name is called glycogen primer. Glycogenin becomes a glycogen primer once 7 glucose residues are attached. Now we have a lot of UDP glucose in the circulation. So what happens? That will be acted upon by the enzyme glycogen synthase and will get added to the glycogen primer. In straight chains, when they get added, you need the linkage 1,4. 1,4 in straight chains. And it will keep on adding until you have 11 glucose residues long. Once you have 11 glucose residues long, still if you keep on adding the glucose, it's going to become a straight chain. It can extend up to kilometers. So you need to restrict it. So you need to branch it. For that, another enzyme comes, branching enzyme. Branching enzyme will help in the formation of branch points, that is alpha 1,6 linkage. So this branching enzyme will take these glucose units, at least 6 glucose residues are attached in 1,4 linkage. It will transfer it to a neighboring chain and establish a branch point at 1,6 linkage. This is how this cycle gets repeated. Again, glycogen synthase, a lot of UDP glucose comes, glycogen synthase, 1,4, 6 residues, once 11 residues are long, the branching enzyme comes and then it transfers the neighboring chain. Again, it establishes a branch point. Then again, more UDP glucose, glycogen synthase enzyme, branching enzyme. Again, the cycle keeps on repeating. And that is how glycogen is formed as a highly branched structure. This is the pathway of glycogen synthesis. Now, this is formed. How do we break it down? Glycogenolysis or glycogen breakdown. So, glycogen has straight chains 1,4 linkage and branch points 1,6 linkage. Here, two major enzymes. Glycogen phosphorylase is the first enzyme. It will break the 1,4 straight chain linkages are all broken by this enzyme. So, it releases glucose as glucose 1 phosphate. Here, one major key point is PLP is a coenzyme for glycogen phosphorylase. This is the only enzyme of carbohydrate metabolism which requires vitamin B6 or its coenzyme form pyridoxal phosphate. Now what happens? It keeps on cleaving the 1,4 linkages. It will cleave until there are 4 glucose residues away from the branch point. So once nearing the branch point, when there are around 4 glucose residues, another enzyme comes into action, glucan transferase. Glucan transferase. it transfers 3 units of glucose, that is a trisaccharide, from this unit to this unit in the straight chain in 1,4 linkage, and it will expose this particular 1,6 point of glycogen. So once it is exposed for action, another enzyme comes, alpha 1,6 glucosidase. This will come and cleave this particular 1,6 glucose, and this is liberated as free glucose. Now, once again the glycogen phosphorylase comes, it cleaves all the glucose in straight chains as glucose 1-phosphate as sequentially releases it. So now this glucose 1,phosphate will be acted upon by phosphoglucomutase to form glucose 6-phosphate. These steps will keep on occurring again and again until the glycogen, which is highly branched, is broken down. Here one key feature is this glucan transferase and alpha-1,6 glucosidase enzyme activities are present in a single enzyme, which is called the debranching enzyme. So these two enzyme activities, glucan transferase and 1,6 glucosidase, are present in only one enzyme, which is called the debranching enzyme. So when there are two enzyme activities inside one enzyme, it is called a tandem enzyme. So this debranching enzyme is a tandem enzyme. Now this process keeps repeating itself, and that is how the complete glycogen molecule gets broken down. Now we have glucose 6-phosphate. At the end, the last step differs in the liver and the muscle. In the liver, you have the enzyme glucose 6-phosphatase, which will break the glucose 6-phosphate to glucose. Whereas in the muscle, you do not have this enzyme, so glucose 6-phosphate will stay as such. So you have a MCQ here. Muscle glycogen does not contribute to blood glucose directly because of the absence of the enzyme glucose 6-phosphatase. So muscle glycogen contributes only up to glucose 6-phosphate. It will not contribute directly to blood glucose. So this summarizes both the pathways of glycogen synthesis or glycogenesis and glycogenolysis or glycogen breakdown.